Okay, this is part 2 of chapter 11. So we're going to talk about what causes evolution, so natural selection, mutation, genetic drift, and gene flow, and how each evolutionary force can influence allele frequencies in the population. So we're really going to talk about this allele frequencies in the population. Okay, so let's go on. All right, so when we talk about allele frequency, what do we really mean? So the first idea you want to remember is that the smallest unit that's able to evolve is going to be a population. So you as an individual, you are not evolving because you have to have changes in your allele frequency, which we'll talk about, which is not possible. Um, so only offspring can be different than you, and any evolution that happens has to happen with a population so that you can have sexual reproduction and you can have the changes in allele frequency. So what is the allele frequency and what is a gene pool? Okay, a gene pool is all the alleles that exist within a population. So if you're thinking about maybe there's plants on an island and those plants have different alleles for different traits that they have, right? If you just took all their alleles in a bucket and you just counted them up, that's the allele frequency. So let's look at this picture here. So there's a little bit of math involved. So here's our phenotype, here's our genotypic frequency, and the total number of individuals. So if you had big Y, and the genotypic frequency is 0.49, that's basically saying that 49% out of this 500 total individuals would have the big Y allele or the genotype. It means 42% would have the heterozygous phenotype, and then 9% would have the homozygous recessive phenotype. Okay, so when you're working with allele frequencies, you want to remember there's an equation, okay? The equation's right here. So P is going to represent the dominant allele, and Q is going to represent the recessive allele. And these are going to be in decimal points, but they always have to equal 1. So in this example here, where it says Mendel's pea flowers had 2 alleles, purple is dominant and Q, white, is recessive, right? So they have to equal 1. So if purple is 0.7, so their allele frequency is 0.7, or 70% of the population has that allele, then you know that the frequency, or how much this little Q allele pops up, has to be 0.3 because they equal 1, right? So this is 0.7, 0.7 plus, let's put a point there, 0.3, right? This equals 1. So if you know one allele frequency, you have to know the other, right? Because 1 represents like 100%. Okay. All right. When you do analysis of the gene frequencies, what you do is a Punnett square. Oops, sorry about that. A Punnett square, but you're going to put the allele frequencies next to it, and then you're going to multiply, right? So P, that's P squared, is 0.49. Q is 0.3, P is 0.7, so PQ, right, a heterozygous individual is 0.21. Same thing here, right? And then the Q squared, 0.3 and 0.3 is 0.9, 0.09, all right? So then you get their frequencies for these predicted offspring, okay? So it's just a number that they put on to a Punnett square. And you just need to keep in mind that the allele frequency P plus Q is 1, and they can figure out the other allele. So if I said the dominant allele is 0.4, okay, equal, which is basically saying 40% of the population, then what is Q, right? Q is going to be 0.6, right? So that's how you do that. All right, so Hardy-Weinberg equation, the Hardy-Weinberg equilibrium, okay, so remember this, it's genetic equilibrium. If a gene pool is stable from one generation to the next, the population is not evolving because the gene pool is constant. Okay, so let's say, so in this case, 2p squared, this is going to be the homozygous dominant, right? PP. And then this is going to be the heterozygous, right? PQ. and then this is going to be the homozygous recessive. All right, so in this quadratic equation, it also equals 1. When you look at this example here, this is just an illustration of how Hardy-Weinberg equilibrium is calculated. So let's say that we have this parent generation, and we have, these are the number of individuals. So we have genotypes homozygous dominant, we have a heterozygous, and we have homozygous recessive, okay? And then these are the genotypic frequencies, okay? So when that works out to what each allele, so the dominant allele is 0.6 and the recessive allele is 0.4. Okay, so you don't have to do the math for that, but I just want to show you that this represents the allele, the genotype frequency. What's the frequency of homozygous dominant? About 36%. What's the allele frequency, I'm sorry, genotypic frequency of a heterozygous? 58%. Sorry, 48%. And then here 16%. So what's calculated for you here is just how frequent do you see the big A allele in this population? You see it's 60%, right? So 60% of the population has a big A allele, 40% of the population has a little a allele. When you do the cross with your allele numbers in place and you do the math, the multiplication, look what happens to the offspring's genotype. Okay, the offspring has 0.36 big A, oh, the same as this. 0.48 big heterozygous, same as that. Okay, and then 0.16, that's the same also. Okay, when you get the same allele frequency from the parent generation to the offspring generation, has anything changed? No, right? The gene pool, exactly how much of each genotype, exactly how much of each allele that's present is exactly the same. So if it's exactly the same, evolution did not happen, because evolution depends on the population scale, a change in the frequency, right? If you change the frequency to the next generation, now you have some evolution happening. So this is kind of a hypothetical, right? So this is basically the standard. Hey, if we see 0 allele frequency change over a generation, there's no evolution occurring. Okay, here's an example of evolution occurring by a change in allele frequency. Okay, so this is the peppered moth example. This is the picture at the beginning of the chapter. So this is a famous example of, it's called industrial melanism, and there is a moth, right? The population of moths, they come in 2 colors. There's the white colored moth and the darker colored moth, and these are called peppered moths. So before the Industrial Revolution, we're talking about history, and the Industrial Revolution was about creating factories. Those factories early on would depending on coal, fossil fuels, so they're very dirty. And before the Industrial Revolution, the birch trees had white bark. And so if you are a white moth before the Industrial Revolution, you would be harder to see, and the birds would pick off the dark ones because the dark ones really stood out against the white background. After the Industrial Revolution, the barks of the trees turned black because of the soot. And then guess what? The white moths turned out to be the ones that stick out. So after the Industrial Revolution, all of a sudden we have an uptick in the darker allele, right? So there's an allele frequency change. First, the allele for white was more predominant because it was protective, and so more of that allele was in the population. Whereas after the Industrial Revolution, we have the dark allele being more prominent because it conferred survivability, right? You could survive these darker tree bark because you blended in better. Okay, so we see there's an allele shift. So the frequency of alleles in this particular population went from white as more frequent to dark as more frequent. So this is going to be evidence of evolution. There's some change happening to the population, right? So the population level, you have a lot more black moths now afterwards. All right, there are other ways that you can change allele frequency, and this one's called genetic drift. And I want you to remember that there are some random processes that can change the allele frequency, and then there's some non-random. So this is random. So what genetic drift is elimination of allele by population just by chance. Just if this was the beginning population you have and you see the allele frequency where the dominant allele is 0.5, the recessive allele is also 0.5, so 50-50. So if you counted each A, big A and little a, you'd see that they're the same. Then out of these, there's going to be 5 rabbits that reproduce. Okay, when they reproduce, here's your second generation. It turns out that we lost a white rabbit, right? So we reduced the number of white rabbits, which means that the allele frequency changed. So now we have 70% with the dominant allele, 30% with the recessive allele. If you mate again, right, just random mating, we get an allele frequency change again. Uh-oh. So now there's 100% dominant alleles and 0 for the recessive allele. So you can imagine that if this goes on again, any of these, it doesn't matter. We've completely out-selected that little a. So big A is the only allele that's going to exist in this population. And we lost the little a allele. So this is genetic drift. And it's basically just random mating that can lead to this change in allele frequency, which is literally the definition of evolution occurring, but at this small level, at the population level. This is a slide which shows that this genetic drift, this random mating, is going to have more an effect on small populations than on big populations. So let's look at this small population of frogs, right? So the allele frequency is half and half for the red and green allele. Random occurrence of random events means that over 5 generations, look what happened to the alleles. It turns out that the green alleles are slowly disappearing for some reason. And so we have a shift in allele frequency. And then on the 10th generation, we have a big shift in allele frequency. Okay, so for some reason, they're not mating with these green frogs. The green frogs are sitting at the wall at the dance, right? So this is a small change. This is a small population. We can see a really big change because we went from 0.5 to 0.1, so definitely a big change. But if you have a bigger population, right, if you have 50 individuals, then that random mating, if you go across the screen here, you can see we shifted a little bit and we shifted a little bit, right? So the population, the allele frequency is more stable if you have a bigger population, and you can have really big changes in the allele frequency if you have just a small population. So hopefully that makes sense. Smaller populations always feel the change of genetic drift a lot more than larger populations because there's more, the gene pool is bigger, and you have more resistance to losing that one allele. All right, so this goes under the genetic drift. So this is also random. It's called the bottleneck effect. And this is where some kind of catastrophe reduces the genetic variability within a population. So in this picture, they like to represent the bottleneck effect as a bottle, and then you're pouring out some alleles. So this represents the different alleles in that population. So you see there's red, there's green, there's pink. So there's a mix of different alleles in the population, but after this catastrophe, it could be a fire, it could be a flood, it can be famine, whatever that catastrophe is, only a few will survive, and those few will have a limited gene pool, right? You're losing a lot of the variation in that gene pool, and now you can see that we've lost the red completely. And so after a bottleneck effect, the gene pool gets smaller, and that is unhealthy for a population because that means that the population are more alike. And so they're going to be it's going to be difficult for them to survive adverse events because they're very similar to each other. Here's another example of bottleneck effect where we have some allele frequencies. So all these different color dots represent alleles. So you can see they're all at 20%. With the 5th generation and 10th generation, with a bottleneck effect, you're reducing the gene pool by a lot, right? We've lost a lot of individuals. You can see here that we've drastically changed the frequency of alleles. And then here in that subsequent population, right after a bottleneck, we have lost the yellow allele, we've lost the pink allele, we have mostly the orange allele, and then just 0.2 and 0.1. So this causes that population to be more and more similar, right? Individuals don't have as many variation of genes, right? They don't have as many alleles present to make them different, so they become more similar. And so they're at risk for anything because there's not a lot of variation in that population, right? It's kind of like having clones, not as close as having clones, but it's not a healthy, it doesn't cause like a healthy population. This very robust gene pool is a healthy population. Okay, the next concept is called gene flow, and this is also random because gene flow is basically where you have two individuals or the species in two places, they'll migrate in, right? So it's all about this. You can think of it as migration. Okay, migrate. And so when an individual migrates in to a different population, they're going to carry new alleles. Okay, and so they're going to introduce some new alleles into this population, right? So the brown allele is going to be introduced into the green population. What does this eventually do for these two groups of beetles, right? It's going to make them a little bit more similar because once you get the brown allele in here mixed with the green, you're going to get maybe some brown offspring. And so they're going to look more similar. You can think about this with, I like to say, it's like human travel. So before boats or planes or any means of like really long distance travel, people that lived in Japan completely were Japanese, right? And people that lived in Sweden were completely pale and they're Swedish. But now that we have a way to travel across the globe, you can have a person from Japan marry a person from Sweden, right? That's basically gene flow. We're introducing these genes from Japan and from Sweden together, and we get something that's more similar. Okay, so the divide between Swedish people and Japanese people, you're going to have a hybrid, right? So you're going to actually make those two populations more and more similar by this gene flow. Okay, we're introducing new genes. All right, the third point is natural selection. This is the only process that's not random in terms of evolution or allele frequency change, because we know it's not random because nature selects, right? So there's definitely a force that's going to push the population in one direction or another, and that's nature, right? So take a look at this chart. We have mice that can come in different variations of fur color. Okay, so this original population has fewer in the extremes and kind of a lot of mice in this intermediate color. Then you can have natural selection. Natural selection is going to depend on like what mouse is best fit. So in this example here, it turns out that the lighter mice don't survive. They're missing, and you get a shift over to the darker colors. This might be because they stick out to owls at night, right? And the owls have picked off the lighter ones and the darker ones survive. So we're changing the allele frequency in a particular direction, and that's going to be for the darker mice. In this example, we have what's called a disruptive selection, where all the intermediates, the ones in the middle, die out, right? They're not fit to survive, but the very, very light and the very, very dark are doing well. Okay, so they're going to be selected for. So we have a shift from this bell curve to the extremes because of what's going on in the nature. Stabilizing selection selects for that middle point. So the mice on the ends, maybe they're easier to spot by certain prey, and this brown mouse maybe blends into the environment better, and we were selecting for that brown color. So in each of these examples, or pushing the allele frequency in one direction or the other. It can be in one direction, it can be at the extremes, and it can be selecting for that middle. All right, so those are the three: directional, disruptive, and stabilizing. I have an example here. This is the oyster shell color. This is a real example. So for oysters, if you have a light shell, they blend in with sandy, shallow water to hide from hungry fish and birds. If you have a dark shell, you hide well in the dark shadows of deeper water. If you have a medium shell, you stand out against both light and dark, so predators can easily spot and eat them. Okay, so if you look at this, which one does this most resemble? Where you have light and dark protected, but the medium goes away. All right, so that would be, if you've guessed, disruptive, because you're selecting for the light and the dark, and the medium ones have gone away. All right, then a fun one here is sexual selection. So natural selection includes sexual selection, which is basically like nature is selecting for those that are best fit to reproduce, but in a way that is behavioral or what they look like. So it's basically the female male, like how do you persuade a female to mate with you? There are very picky females out there, right? So individuals, certain traits are more likely than others to obtain mates. That's what sexual selection is for. We see differences because of this in males and females. Females tend to be always the choosy ones. Males tend to exhibit traits that makes them more likely to win the female. So either they have really brightly colored feathers, as in birds, they have a very specific dance they need to do, or they have large horns, or they're just bigger and stronger to compete for a mate. Okay, so in the peacock example, Darwin did say that he hated this bird because he didn't understand why he had such a big tail. Right, because he said, oh, the tails are so heavy that they can't even fly very well. So that's bad because they can't get away from predators. Their tail is also super bright. So it's not like they can hide from predators. They're really easy to spot. So those are questionable. Like, why would you have this tail? Oh, because the females, the peahen, is loving this big tail, right? So the bigger and flashier your tail is, the peahens will mate with you. So when you have this really big advantage, right, your genes are going to get passed on to the offspring. So this is sexual selection. And then this one is a really fun video based on a dance that I'll show you. Okay, so the question for you is, which choice below is not a random process for population evolution? We forgot a word here. Is it gene flow, genetic drift, bottleneck effect, or natural selection? Well, these, oops, sorry about that. Gene flow, genetic drift, and bottleneck effect, these are all random occurrences that would change the allele frequency. and only natural selection is not random.